Hello students, this is Module 4.2.3, Biotechnology Techniques. On the slide in front of you, you can see some of the types of biotechnology or some of the techniques which are available. This module will cover the first three, then we'll have another module to talk about cloning, which is a little more extensive. The first of the biotechnology techniques we'll discuss is called selective breeding. Selective breeding is simply when humans select a parentage to get the desired offspring of other species. This is especially common in agriculture, where we'll take plants and breed them in order to get better yields, or we'll breed animals to get either behavior or anatomy that we like. For example, more docile cows, or cows with more muscle, more meat, or cows that give more milk, for example. All these are examples of selective breeding. This is also known as, here it says, artificial selection. And the example that's shown here on the slide shows that the wolf gave rise to all the many breeds of domestic dogs through selective breeding. And you can see by the great variety of breeds of domestic dogs that this is a very powerful technique. And it takes multiple generations to get offspring with the traits that we desire, but it is often very, very effective. And this is really the old type of biotechnology. This has been around for many thousands of years, at least 10,000 years, and probably longer if we go back to some of the domestication of dogs that served as hunting companions when we were still hunter-gatherers. Agriculture started in a substantive way about 10,000 years ago, and then, of course, we were using selective breeding for many different species. Here's another slide that illustrates some examples of selective breeding. There's the wolf giving rise to all these varieties of dog. It is interesting to note that a Chihuahua and a Great Dane are the same species and are interfertile. It's just if a male Chihuahua is going to mate with a female Great Dane, he's going to need a ladder or something. Other examples include chili peppers. There were just a couple of species, and we've bred many. I don't know how many of you are familiar with the chiltepines, these little tiny ones here, but they're very excellent in salsa. And they were very small, and they actually pointed upward because they were dispersed by birds. Later, when we bred chili peppers to be really large, they had to hang down. Another example is that we used a single ancestor mustard plant to give rise to a variety of vegetable crops by selecting for different attributes of the mustard plant. For example, when we selected for flowers and stems, this gave rise to broccoli. When we selected for flower clusters, this gave rise to cauliflower. When we selected for the terminal bud, just the vegetative growth right at the tip of the plant, this created cabbage. If we select for lateral buds, this produces Brussels sprouts. We select for the leaves, this produces a crop called kale. And we select for the stem, and this gives us kohlrabi, which I've never had but understand is supposed to be pretty good. I think I had someone tell me that they use it in Hungarian food. I really don't have first-hand experience with it. So let's leave the old pre-technological example of biotechnology and look at something that has perhaps a little bit more technology in it. And this is what we've already talked about. This is simply recombinant DNA, or genetic engineering. And when we say recombinant DNA, it's combining DNA from two sources. And so here it's defined as the manipulation and combination of DNA from different sources, also known as gene splicing. And we already discussed the tool that we use, the mechanism. These are restriction enzymes. We cut two sources of DNA with the same restriction enzyme, we get complementary sticky ends, and they will go together, and we can simply use DNA ligase to hook the little spliced bits together. And a great example of this would be putting the gene for human insulin production into bacteria. Probably most of you know someone that has diabetes, and the fact that we're able to now manipulate genes allows us to take the gene for human insulin production, put it into bacteria using a number of the tools that we've talked about. We use both restriction enzymes and plasmids, and we put this gene into bacteria, and we can make as much human insulin as we need to. It used to be that if someone had diabetes, they would die. And then we figured out that diabetes was caused by a lack of insulin, and so then we actually tried using insulin from other animals. For example, we'd take insulin from pigs in the slaughterhouse, and that's what diabetics used for quite a while. And it worked okay unless the individual had a very sensitive immune system, and then their immune system would pick up the foreign proteins in the pig insulin, and then they would die because they don't have a way to supply insulin that their body doesn't produce enough of. And finally, now we live in an era of biotechnology where we can produce as much human insulin as we need with no risk of rejection by the immune system. It's really quite amazing. So that's just one application of biotechnology, but it's an important one. We will discuss more examples of this, recombinant DNA or gene splicing technology and all that it can do for us. Here's a couple of examples that when we were first pioneering these techniques that scientists were very excited about. They took a gene from a firefly for an enzyme called luciferase, so the gene to make luciferase. Luciferase actually allows the bioluminescence, the production of light. And we put that gene into mice, so we had glowing mice. We put the gene into fish. These are zebra danios, put it in there. And then we had glowing fish. They put a similar gene into a tobacco plant. And so we can see some, these are frivolous in terms of doing any good, but they were an opportunity for biotechnologists to see firsthand the impacts that their experiments were having. And so they could see if an experiment was successful or not. And so that's why they chose a gene such as this. Okay, let's look at a third example of techniques of biotechnology. This one is called DNA fingerprinting. When we were talking about human ABO blood types, I mentioned that they formerly used these blood types to try to determine paternity or to determine whether or not someone could be ruled out as a suspect in crimes where biological evidence, either blood or semen or saliva, was left. But of course, I'm sorry, that's not going to work very well, where blood was left only. The others, I'm talking about DNA. And of course, that's a technique that replaced human ABO blood typing for identification, was the technique here, DNA fingerprinting. This is when we're using the uniqueness of the genetic structure of an individual to identify a sample to a particular person. And so this is really quite definitive in terms of matching a sample to an individual. It turns out that the majority of DNA is the same. You learned this in the NOVA video, Cracking the Code, that they said 99% of human DNA is the same. And I think it's a little bit less than that we found since that video was made. But nevertheless, the vast majority of human DNA is almost identical. And so we have to focus on the part that is different. And we actually use non-coding sequences that have multiple copies. And this is the area that we focus on for DNA fingerprinting. Here's the steps of DNA fingerprinting. So we take some sections of the variable DNA from two DNA sources. That would be from a sample left at a crime scene and from a suspect. Or if we're trying to do paternity, we'd take it from a child and from a potential father. We'd cut that DNA using an assemblage of restriction enzymes, and they use the right enzymes to cut in these variable areas. Then we would use a technique called gel electrophoresis. And gel electrophoresis, essentially, you make a little slab of Jell-O with some little whales in it, and you put in the little whales or little openings, you put the DNA samples, and then you electrify the gel. This is done under liquid in what is called a buffer solution. And the smaller snippets of DNA, the smaller sequences of DNA are able to move through the gel more rapidly than the larger ones. And so, in other words, the gel electrophoresis separates the DNA segments from after they were digested with restriction enzymes by size. And so this produces a unique pattern of bands, because where more DNA fragments end up, you get a band. And we actually use some technique to visualize the bands, usually a dye, but we can also use radioactivity. But if the DNA is from the same, if the two sources of DNA match, then they're from the same person, and the banding pattern will be the same. Otherwise, you'd see different banding patterns. This actually shows gel electrophoresis, so they have this little kind of plexiglass plate. You can see the electrodes. DNA is negatively charged, and so it will move in an electrical current produced by these. This person here is loading the gel with a pipette, so putting it into the little wells at the end. And the result of running the gel after in a DNA fingerprinting experiment, oh, here's another gel. This one's kind of wanky, you see, but it's already loaded, and you can see that the DNA has been dyed blue so that we will be able to visualize it or see it. And here is a gel. Now take a look at this gel. This is in the center. We have, it looks like 8 lanes in this gel, and the one here in the center is from the blood at the crime scene. And which one of these numbers of suspects better get themselves a good lawyer? Well, if you look closely, you can see without difficulty that it better be number 3. And we can see that the bands match up. I just put a few arrows in. I could have put a lot more because the banding is identical top to bottom. And so you can see how each individual produces a unique pattern of bands. Here is oh, there we go. DNA, you're an excellent and objective witness. And here is the DNA from a famous case. If you look over here at the side, you can see from the boot, from the foyer, and this is at Bundy Street, and that's O.J. Simpson, Nicole Brown, and Ron Goldman. This is when O.J. Simpson pretty much got away with murder, it would seem, at least according to the DNA. The reason he was acquitted was because his lawyers claimed that the DNA had been contaminated and that the Los Angeles Police Department had intentionally tried to frame him. But the DNA samples were unequivocal in matching O.J. Simpson, so he was definitely there according to the blood at the scene. And it just shows you that a good lawyer is something you need in the American justice system, I suppose. In any case, that will conclude this module.